Hi class, this is your instructor, Skylar Huff, and we're now in Chapter 5. This chapter is about membranes. A cell's interactions with the environment are critical, a give and take that never ceases. Without it, life could not exist. Living cells are encased within a lipid membrane through which few water-soluble substances can pass. This plasma membrane also contains protein passageways that permit specific substances to move into and out of the cell and allow the cell to exchange information with its environment. Eukaryotic cells also contain internal membranes like those of the mitochondrion and the endoplasmic reticulum, as you see pictured here. We call the delicate skin of lipids with embedded protein molecules that encase the cell a plasma membrane. So let's examine the structure and function of cell membranes. So from there we'll move on to phospholipids, proteins, and then we'll end things with the way in which passive transport occurs, including active transport, and then end the chapter with bulk transport by way of endo and exocytosis. The membranes that encase all living cells are two phospholipid sheets that are only 5 to 10 nanometers thick, and more than 10,000 of these sheets piled on one another would equal just the thickness of the sheet of paper in your textbook. Biologists established the components of the membranes, not only lipids, but also proteins and other molecules through biochemical assays, but the organization of the membrane components remained elusive. We begin by considering the theories that have been advanced by the structure of the plasma membrane. We then look at the individual components of the membranes more closely. So the lipid bilayer that forms the foundation of a cell's membrane is a bilayer formed of phospholipids. These phospholipids includes primarily the glycerol phospholipids, as you see in figure 5.1. Note the sphygmolipid such as sphygmomyelin, and you can see sphygmomyelin in figure 5.2, which your textbook shows you right here. So if you look closely, you will see those there. So note, although these look superficially similar, they are built on a different carbon skeleton. For many years, biologists thought that protein components of the cell membrane covered the inner and outer surfaces of the phospholipid bilayer like a coat of paint. An early model portrayed the plasma membrane as a sandwich, a phospholipid bilayer between two layers of globular protein. In 1972, S. Jonathan Singer and Garth J. Nicholson revised the model in a simple but profound way. They proposed that the globular proteins are inserted into the lipid bilayer, with the nonpolar segments in contact with the nonpolar interior of the bilayer, and their polar portions protruding out from the membrane surface. In this model, called the fluid mosaic model, the mosaic of proteins floats in or on the fluid lipid bilayer like boats on a pond. So if you look at figure 5.3, it shall show you that, though it says figure 5.2 here, this is figure 5.3, showing you how they float as if it were a boat. So that mosaic of glycoproteins, the integral proteins, the glycolipids, or even the cholesterol. We now recognize two categories of membrane proteins based on their association with the membrane. The integral proteins are associated by being embedded within the membrane. I'll say again, integral membrane proteins are embedded in the membrane, whereas peripheral proteins, they are associated with that surface of the membrane. So peripheral proteins, surface membrane proteins. Cellular membranes consist of 4 component groups. So a eukaryotic cell contains many membranes. Although they are not identical, they share the same fundamental architecture. Cell membranes are assembled from 4 components, and your textbook has a table for this. It's called Table 5.1. Please see Table 5.1. Phospholipid bilayer. Every cell membrane is composed of phospholipids in a bilayer. The other components of the membrane are embedded within the bilayer, which provides a flexible matrix and at the same time imposes a barrier to permeability. Animal cell membranes also contain cholesterol, a steroid with a polar hydroxyl group too. And then plant cells have other sterols but no cholesterol. Transmembrane proteins. A major component of every membrane is a collection of proteins that float in the lipid bilayer. These proteins have a variety of functions, including transport and communication across the membrane. Many integral membrane proteins are not fixed in position. They move about just as the phospholipid molecules do. Some membranes are crowded with proteins, but in others, the proteins are more sparsely distributed. Interior protein network. Membranes are structurally supported by intracellular proteins that reinforce the membrane shape. For example, a red blood cell has a characteristic biconcave shape because of a scaffold made of a protein called spectrin that links proteins in the plasma membrane with actin filaments in the cell's cytoskeleton. Membranes use networks of other proteins to control the lateral movements of some key membrane proteins, anchoring them to specific sites. Cell surface markers. As you learned in Chapter 4, membrane sections assemble in the endoplasmic reticulum, transfer to the Golgi apparatus, and then are transported to the plasma membrane. The endoplasmic reticulum adds chains of sugar molecules to the membrane proteins and lipids, converting them into glycoproteins and glycolipids. Different cell types exhibit different varieties of these glycoproteins and glycolipids on their surfaces, which act like cell identity markers. So if you look at Table 5.1, it shows you each of those that we just went through, along with its composition, function, how it works, and an example. Originally, it was believed that because of its fluidity, the plasma membrane was uniform, with lipids and proteins free to diffuse rapidly in place of the membrane. However, in the last decade, evidence has accumulated suggesting the plasma membrane is not homogeneous and contains microdomains with distinct lipid and protein composition. This was first observed in epithelial cells, in which the lipid composition of the apical and basal membranes was shown to be distinctly different. Theoretical work also showed that the lipids can exist in either a disordered or an ordered phase within the bilayer. Electron microscopy allows biologists to examine the delicate filmy structure of a cell membrane. We discussed 2 types of electron microscopes in Chapter 4, the transmission electron microscope and the scanning electron microscope. Both provide illuminating views of membrane structure. And you're seeing here an electron micrograph of adjacent cell membranes. The micrograph shows 2 adjacent cells. The structure of the plasma membrane of each cell is clear, and the micrograph has been false colored to emphasize the bilayer nature of the membranes. This is figure 5.4. Lipodomics. Lipodomics is a field defining the number of biological function of lipids is revealing a significant diversity of membrane lipids. Although there are over 1,000 distinct lipids defined in cells, we can organize them into only three classes: glycerol phospholipids, sphingolipids, and sterols such as cholesterol. The classical phospholipid bilayer consists of a combination of glycerol phospholipids and sphingolipids. The glycerol phospholipids are the most diverse, with head groups that can have both positive and negative charge, or primarily negative charge. Phospholipids can also vary in length and composition of a fatty acid tail, with it being either saturated or cis unsaturated. Sphingolipids usually contain saturated hydrocarbon chains. These components are not uniformly distributed in biological membranes and different cellular compartments have distinct membrane lipid composition, as discussed later in this very same section. So here you have phosphodactyline. Phospholipids spontaneously form bilayers because of their amphipathic structure. The polar head groups are hydrophilic, whereas the nonpolar hydrocarbon tails are hydrophobic. The two nonpolar fatty acids extend in one direction. Roughly parallel to each other, shown before you now in figure 5.7. The polar phosphate group points in the other direction. So there you have it. To represent this structure, phospholipids are also often diagrammed as polar head with two dangling non-polar tails. What happens when a collection of phospholipid molecules is placed in water? The polar water molecules repel the long nonpolar tails of the phospholipids while seeking partners for hydrogen bonding. Because of the polar nature of water molecules, the nonpolar tails of the phospholipids end up packed closely together, sequestered as far as possible from the water. Every phospholipid molecule is oriented with its polar head toward water and its nonpolar tails away. When two layers form, the tails facing each other, no tails ever come in contact with the water, the resulting structure is the phospholipid bilayer. Phospholipid bilayers form spontaneously, driven by the tendency of water molecules to form the maximum number of hydrogen bonds. The nonpolar interior of a lipid bilayer impedes the passage of any water-soluble substance through the bilayer, just as a layer of oil impedes the passage of a drop of water. This barrier to water-soluble substances is the key biological property of the lipid bilayer. A lipid bilayer is stable because water's affinity for hydrogen bonding never stops. Just as surface tension holds a soap bubble together, even though it's made of a liquid, so hydrogen bonding of water holds a membrane together. Although water drives phospholipids into a bilayer configuration, it does not have any effect on the mobility of phospholipids and their non-lipid neighbors in the bilayer. Because phospholipids interact relatively weakly with one another, individual phospholipids are unanchored, and unanchored proteins are comparatively free to move about within the membrane. This can be demonstrated vividly by fusing cells and watching their proteins intermix with time. The degree of membrane fluidity changes with the composition of the membrane itself. much like triglycerides can be solid or liquid at room temperature depending on their fatty acid composition. Membrane fluidity can be altered by changing the membrane's lipid composition. Glycerol phospholipids that are saturated or monocis unsaturated tend to make the membrane less fluid as they pack well. Similarly, sphingolipids, which are usually saturated, also make the membrane less fluid. Changes to the environment that have drastic effects on the plasma membrane of single-celled organisms such as bacteria. Increasing temperature makes the membrane more fluid, and decreasing temperature makes it less fluid. Bacteria have evolved mechanisms to maintain a constant membrane fluidity despite fluctuating temperatures. Some bacteria contain enzymes called fatty acid desaturases that can introduce double bonds into fatty acids in membranes. And genetic studies involving either inactivation of these enzymes or introduction of them to cells that normally lack them indicate that the action of these enzymes confers cold tolerance. At colder temperatures, the double bonds introduced by the fatty acid desaturase make the membrane more fluid, counteracting the environmental effect of the reduced temperature. Although most lipids are synthesized in the endoplasmic reticulum, the composition of the endoplasmic reticulum membrane, the Golgi stack, and the plasma membrane are quite distinct. These differences in lipid composition affect the structure and function of each membrane compartment. Cell membranes contain a complex assembly of proteins enmeshed in the fluid soup of phospholipid molecules. This very flexible organization permits a broad range of interactions with the environment, some directly involving membrane proteins. Although cells interact with their environment through their plasma membranes in many ways, we will focus on 6 key classes of membrane protein in this chapter and later on in Chapter 9. Transporters, enzymes, cell surface receptors, cell surface identity markers, cell-to-cell adhesion proteins, attachments to the cytoskeleton, and effect membrane structure. Transporters. Membranes are selective, allowing only certain solutes to enter or leave the cell through either channels or carriers composed of proteins. Enzymes. Cells carry out many chemical reactions on the interior surface of the plasma membrane using enzymes attached to the membrane. Cell surface receptors. Membranes are exquisitely sensitive to chemical messages, which are detected by receptor proteins on their surfaces. Cell surface identity markers. Membranes carry cell surface markers that identify them to other cells. Most cell types carry their own ID tags, specific combinations of cell surface proteins and protein complexes such as glycoproteins that are characteristic of that cell type. Cell-to-cell adhesion proteins. Cells use specific proteins to glue themselves to one another. Some act by forming temporary interactions, and others form a more permanent bond. Attachment to the cytoskeleton. Surface proteins that interact with other cells are often anchored to the cytoskeleton by linking proteins. Proteins that affect membrane structure. Membranes tend to form spheres or sheets in aqueous solutions. Wedge-shaped proteins can cause membranes to bend, allowing the formation of tubes and folded sheets. As just detailed, membrane proteins can serve a variety of functions. These diverse functions arise from the diverse structures of these proteins, yet they also have common structural features related to their role as membrane proteins. Some membrane proteins are attached to the surface of the membrane by special molecules that associate strongly with phospholipids. Like a ship tied to a floating dock, these anchoring proteins are free to move about on the surface of the membrane, tethered to a phospholipid. The anchoring molecules are modified lipids that have non-polar regions that insert into the internal portion of the lipid bilayer, and chemical bonding domains that link directly to proteins. So those are the anchoring molecules. Cell membranes contain a variety of different transmembrane proteins, which differ in the way that they traverse the lipid bilayer. The primary difference lies in the number of times that the membrane protein crosses the membrane. Each membrane spanning region is called a transmembrane domain. These domains are composed of hydrophobic amino acids, usually arranged into alpha helices. So proteins that actually span the lipid bilayer, known as transmembrane proteins. In other words, the part of the protein that extends through the lipid bilayer and that is in contact with the non-polar interior are alpha helices or beta-pleated sheets that consist of non-polar amino acids. Some transmembrane proteins have extensive non-polar regions with secondary configurations of beta-pleated sheets instead of alpha helices. The beta-pleated sheets, or the beta sheets as they're called too, form characteristic motifs, which folding back and forth in a cylinder, so the sheets arrange themselves like a pipe through the membrane. This forms a polar environment in the interior of the beta sheets spanning the membrane. So this is called a beta barrel. Open on both ends is a common feature of the porin class of proteins found within the outer membrane of some bacteria, the openings allow molecules to pass through the membrane. Bending membranes. So many intercellular membranes have elaborate structure, like the folds of tubular networks of the endoplasmic reticulum. Phospholipid composition can affect membrane shape, but proteins also affect membrane structure in several ways. Wedge-shaped membrane proteins called reticulins will cause the membrane to bend, or if they're enough, to form a tube. Many substances can move in and out of the cell without the cell having to expend energy. This type of movement is termed passive transport. Some ions and molecules can pass through the membrane fairly easily and do so because of a concentration gradient, a difference in the concentration inside the membrane versus outside. Some substances also move in response to a concentration gradient, but do so with specific protein channels in the membrane. Molecules and ions dissolved in water are in constant random motion. This random motion causes a net movement of these substances from regions of higher concentration to regions of lower concentration, a process called diffusion. Net movement by diffusion will continue until the concentration is the same in all regions. Consider what happens when you add a drop of colored ink to a beaker of water. Over time, diffusion of the ink molecules causes them to become dispersed throughout the solution. In the context of cells, we need to consider both the relative concentrations of a substance inside and outside of the cell, and how easily the substance can cross the membrane. The major barrier to crossing a biological membrane is the hydrophobic interior that repels polar molecules, but not non-polar molecules. If a concentration difference exists for a non-polar molecule, it will move across the membrane until the concentration is equal on both sides. At this point, movement in both directions still occurs, but there is no net change in either direction. This includes molecules like oxygen and nonpolar organic molecules such as steroid hormones. The plasma membrane has limited permeability to small polar molecules and very limited permeability to large polar molecules and ions. The movement of water, one of the most important polar molecules, will be discussed separately. So here is that beaker. There is that ink or some solute such as potassium permanganate, and you're seeing from this figure called figure 510. So nothing's there at A, it's dropped in at B. C, it's diffusing. D, it has diffused completely, and they're moving randomly, them being the solute molecules. Many important molecules required by cells cannot easily cross the plasma membrane. These molecules can still enter the cell by diffusion through specific channel proteins or carrier proteins embedded in the plasma membrane, provided there is a higher concentration of the molecule outside of the cell than inside the cell. We call this process of diffusion mediated by a membrane protein, facilitated diffusion. Channel proteins have a hydrophilic interior that provides an aqueous channel through which polar molecules can pass when the channel is open. Carrier proteins, in contrast to channel proteins, bind specifically to the molecule they assist, much as an enzyme binds to its substrate. These channels and carriers are usually selective for one type of molecule, and thus the cell membrane is said to be selectively permeable. In Chapter 2, you saw that ions, by way of atoms, so you saw that atoms with an unequal number of protons and electrons have an electrical charge and are called ions. So again, you saw that atoms with an unequal number of protons and electrons have an electric charge and are referred to as ions. Those that carry a positive charge are called cations. Those that carry a negative charge are called anions. Because of their charge, ions interact well with polar molecules such as water, but are repelled by non-polar molecules such as the interior of the plasma membrane. Therefore, ions cannot move between the cytoplasm of a cell and the extracellular fluid without the assistance of transport proteins. Ion channels. Ion channels possess a hydrated interior that spans the membrane. Ions can diffuse through the ion channel in either direction depending on their relative concentration across the membrane. Some channel proteins can be opened or closed in response to a stimulus. These channels are called gated channels, and depending on the nature of the channel, the stimulus can be either chemical or electrical. So keep in mind that you should know this, and I will expand upon this in Chapter 9. 3 conditions determine the direction of net movement of the ions. 1 would be their relative concentrations on either side of the membrane. Secondly, the voltage difference across the membrane and for the gated channels. And then thirdly and finally, the state of the gate, whether it's open or closed. A voltage difference is an electrical potential difference across the membrane called a membrane potential. Changes in membrane potential form the basis for the transmission of signals in the nervous system and some other tissues. So we discussed this topic in detail in Chapter 42, which is not part of this course, though you will be taught some of that information by way of Chapter 9. Each type of channel is specific for a particular ion, such as calcium, sodium, potassium, or even chloride ions in some cases. For more than one cation or anion, So ion channels play an essential role in signaling by the nervous system. Carrier proteins can help transport both ions and other solutes, such as some sugars and amino acids, across the membrane. Transport through a carrier protein is still a form of diffusion and therefore requires a concentration difference across the membrane. Carriers must bind to the molecule they transport, so the relationship between concentration and the rate of transport differs from that due to simple diffusion. As concentration increases, transport by simple diffusion shows a linear increase in the rate of transport. But when a carrier protein is involved, a concentration increase means that one or more of the carriers are bound to the transported molecule. At high enough concentrations, all carriers will be occupied and the rate of transport will be constant. This means that the carrier exhibits saturation. This situation is somewhat like that of a stadium or the cell, where the crowd must pass through turnstiles to enter. If there are unoccupied turnstiles, you can go right on through. But when all of them are occupied, you must wait. When ticket holders are passing through the gates at maximum speed, the rate at which they can enter cannot increase, no matter how many are waiting outside. So this shows you how facilitated diffusion works. Still going from a higher to a lower concentration. Several examples of facilitated diffusion can be found in the plasma membrane of vertebrate red blood cells. One red blood cell carrier protein, for example, transports a different molecule in each direction. Chloride ions in one direction and bicarbonate ions in the opposite direction. As you will learn in Chapter 47, this carrier protein is important in the uptake and the release of carbon dioxide. Glucose is transported in red blood cells by facilitated diffusion through a specific carrier protein. The chemistry of this is interesting, as red blood cells keep their internal concentration low by phosphorylating glucose as it enters the cell. This has several consequences. Glucose phosphate no longer binds to the transporter and therefore cannot leave the cell. It is the form of glucose that enters glycolysis, and it maintains a steep concentration gradient for unphosphorylated glucose, favoring its entry into the cell. The glucose transporter is a carrier protein that specifically binds to glucose. When glucose binds to that transporter, this alters the shape of the carrier, pulling glucose through the bilayer and releasing it inside the plasma membrane. Releasing glucose causes the transporter to revert to its original shape so it can bind another glucose molecule outside the cell. The cytoplasm of a cell contains ions and molecules such as sugars and amino acids dissolved in water. The mixture of these substances and water is called an aqueous solution. Water is termed the solvent, and the substances dissolved in the water are solutes. Both water and solutes tend to diffuse from regions of higher concentration to ones of lower concentration. That is, they diffuse down their concentration gradients. When two regions are separated by a membrane, what happens depends on whether the solutes can pass freely through that membrane. Most solutes, including ions and sugars, are not lipid soluble and therefore are unable to cross the lipid bilayer. The concentration gradient of these solutes can lead to the movement of water. Water molecules interact with dissolved solutes by forming hydration shells around charged solute molecules. When a membrane separates two solutions with different concentrations of solutes, the concentrations of free water molecules on the two sides of the membrane also differ. The side with the higher solute concentration has tied up more water molecules in hydration shells and thus has fewer free water molecules. As a consequence of this difference, free water molecules move down their concentration gradient toward higher solute concentration. This net diffusion of water across a membrane toward higher solute concentration is called osmosis, and this is shown before you now. The concentration of all solutes in a solution determines the osmotic concentration of a solution. If two solutions have unequal osmotic concentrations, the solution with the higher concentration is hypertonic, and the solution with the lower concentration is hypotonic. When the two solutions have the same osmotic concentrations, these solutions are isotonic, meaning equal. The terms hyperosmotic, hypoosmotic, and isoosmotic are also used to describe these conditions. So there you have those. A cell in any environment can be thought of as a plasma membrane separating two solutions, the cytoplasm and the extracellular fluid. The direction and extent of any diffusion of water across the plasma membrane is determined by comparing the osmotic strength of these solutions. Put another way, water diffuses out of a cell in a hypertonic solution. That is, the cytoplasm of the cell is hypotonic compared with the extracellular fluid. This loss of water causes the cell to shrink until osmotic concentrations of the cytoplasm and the extracellular fluid become equal. The transport of water across the membrane is complex. Studies on artificial membranes show that water, despite its polarity, can cross the membrane, but this flow is limited. Water flow in living cells is facilitated by aquaporins, which are specialized channels for water. A simple experiment demonstrates this. If an amphibian egg is placed in hypotonic, if an amphibian egg is placed in hypotonic spring water, the solute concentration in the cell is higher than that of the surrounding water. It does not swell. If aquaporin mRNA is then injected into the cell, the channel proteins are expressed and appear in the egg's plasma membrane. Water can now diffuse into the egg, causing it to swell. More than 11 different kinds of aquaporins have been found in mammals. These fall into two general classes, those that are specific for water only, and those that allow small hydrophilic molecules such as glycerol or urea to cross the plasma membrane as well. The latter class explains how some membranes allow the easy passage of small hydrophilic substances. The human genetic disease, hereditary diabetes insipidus, has been shown to be caused by non-functional aquaporin protein. This disease causes the excretion of large volumes of dilute urine, illustrating the importance of aquaporins to our physiology. What happens to a cell in a hypotonic solution? That is, a cell's cytoplasm is hypertonic relative to the extracellular fluid. In this situation, water diffuses into the cell from the extracellular fluid, causing the cell to swell. The presence of the cytoplasm pushing out against the cell membrane or hydrostatic pressure increases. The amount of water that enters the cell depends on the difference in the solute concentration between the cell and the extracellular fluid. This is measured as osmotic pressure, defined as a force needed to stop osmotic flow. If the membrane is strong enough, the cell reaches an equilibrium at which the osmotic pressure, which tends to drive the water into the cell, is exactly counterbalanced by the hydrostatic pressure, which tends to drive the water back out of the cell. However, a plasma membrane itself cannot withstand large internal pressures, and an isolated cell under such conditions would burst like an overinflated balloon. And you can see this in figure 5.13 in your text. Accordingly, it is important for animal cells, which only have plasma membranes, to maintain osmotic balance. In contrast, the cells of prokaryotes, fungi, plants, and many protists are surrounded by strong cell walls which can withstand high internal pressures without bursting. So in figure 5.13, it shows you the behavior of human red blood cells and plant cells in each of those three solutions mentioned earlier: the hypertonic solution, the isotonic solution, and the hypotonic solution. You should know how both of these types of cells behave in each of those three types of solutions. Organisms have developed many strategies for solving the dilemma posed by being hypertonic to the environment and therefore having a steady influx of water by osmosis. Extrusion. Single-cell eukaryotes such as the protist Paramecium use organelles called contractile vacuoles to remove water. Each vacuole collects water from various parts of the cytoplasm and transports it to the central part of the vacuole near the surface. The vacuole possesses a small pore that opens to the outside of the cell. By contracting rhythmically, the vacuole pumps out or extrudes through this pore the water that is continuously drawn into the cell by osmotic forces. Isoosmotic regulation. Some organisms that live in the ocean adjust their internal concentration of solutes to match that of the surrounding seawater. Because they are isoosmotic with respect to their environment, no net flow of water occurs into or out of these cells. Many terrestrial animals solve the problem in a similar way by circulating a fluid through their bodies that bathes cells in an isotonic solution. The blood in your body, for example, contains a high concentration of the protein albumin, which elevates the solute concentration of blood to match that of your cell's cytoplasm. And then turgor. Most plant cells are hypertonic to their immediate environment, containing a higher concentration of solutes in their central vacuoles. The resulting internal hydrostatic pressure, known as turgor pressure, presses the plasma membrane firmly against the interiors of the cell wall, making the cell rigid. Most marine plants depend on turgor pressure to maintain their shape, and thus they will when they lack sufficient water. Diffusion, facilitated diffusion, and osmosis are passive transport processes that move materials down their concentration gradients. But cells can actively move substance across a cell membrane up their concentration gradients. This process requires the expenditure of energy, typically from ATP, and is therefore called active transport. Like facilitated diffusion, active transport involves highly selective protein carriers within the membrane that bind to transported substance, which could be an ion or a simple molecule such as sugar, if not sugar, an amino acid, or even a nucleotide. These carrier proteins are called uniporters if they transport a single type of molecule, and symporters or antiporters if they transport two different molecules together. Symporters transport two molecules in the same direction. Antiporters transport two molecules in opposite directions. These terms can also be used to describe facilitated diffusion carriers. Active transport is one of the most important functions of any cell. It enables a cell to take up additional molecules of a substance that is already present in its cytoplasm and concentrations higher than that in the extracellular fluid. Active transport also enables a cell to move substances out of its cytoplasm and into the extracellular fluid, despite higher external concentrations. The use of energy from ATP in active transport may be direct or indirect. Let's first consider how ATP is used directly to move ions against their concentration gradients. So here are those carrier proteins mentioned moments earlier. The uniporters moving one molecule at a time, with symporters moving two molecules in that same direction, and antiporters moving two molecules in opposite direction. More than 1/3 of all energy expended by an animal cell that is not actively dividing is used in the active transport of sodium and potassium ions. Most animal cells have a low internal concentration of sodium relative to their surroundings and a high internal concentration of potassium. They maintain these concentration differences by actively pumping sodium out of the cell and potassium in. The remarkable protein that transports these two ions across the cell membrane is known as the sodium-potassium pump. You can see that in figure 5.14. This carrier protein uses the energy stored in ATP to move these two ions. In this case, the energy is used to change the conformation of the carrier protein, which changes its affinity for either sodium ions or potassium ions. This is an excellent illustration of how subtle changes in the structure of a protein affect its function. Cells use the sodium-potassium pump to create a high concentration of sodium outside the cell and a high concentration of potassium inside the cell. Movement against a concentration gradient can be done only by active transport. I repeat, movement against a concentration gradient can only be done by active transport. Passive transport by diffusion would equalize concentration across the membrane. These gradients of sodium and potassium are important in the creation of an electrical potential across the membrane, critical to the function of nerve cells. This process is: the sodium-potassium pump works through the following series of conformational changes in the transmembrane protein. So this is summarized here before you, and this is the figure shown in your textbook. It says figure 516, so there you go. With ATP bound to the pump, there are 3 sodium that bind to the cytoplasmic side of the protein. The bound ATP is hydrolyzed to ADP, transferring a phosphate group to the pump. The protein is now phosphorylated. The phosphorylated protein undergoes a conformational change, exposing the 3 sodium ions to the outside. In this conformation, the pump now has a low affinity for sodium, allowing those 3 bound sodium ions to diffuse into the extracellular fluid. The phosphorylated protein has a high affinity for potassium, 2 of which bind to the extracellular side of the pump after sodium diffuses away. The binding of potassium causes hydrolysis of the bound phosphate and a conformational change back to the binding pocket facing the cytoplasm. The pump binds ATP, returning to a state of low affinity for potassium and a high affinity for sodium. The bound potassium diffuses away, and 3 new sodium can now bind to initiate another cycle. This is the sodium-potassium pump. In every cycle, 3 sodium leave the cell and 2 potassium enter. The changes in protein conformation that occur during the cycle are rapid, enabling the carrier to transport as many as 300 sodium per second. The sodium-potassium pump appears to exist in all animal cells, although cells vary widely in the number of pump proteins they contain. Some molecules are moved against a gradient by using the energy stored in the gradient of a different molecule. In this process, called coupled transport, the energy released as one molecule moves down its concentration gradient is captured and used to move a different molecule against its concentration gradient. As you just saw, the energy stored in ATP molecules can be used to create a gradient of sodium and potassium across the membrane. These gradients can then be used to power the transport of other molecules across the membrane. As one example, let's consider the active transport of glucose across the membrane in animal cells. Glucose is such an important molecule that there are a variety of transporters for it, one of which discussed in section 5.4 under passive transport. In a multicellular organism, intestinal epithelial cells can have a higher concentration of glucose inside the cell than outside. So, these cells need to be able to transport glucose against its concentration gradient. This requires energy and a different transporter from the one involved in facilitated diffusion of glucose. The active glucose transporter uses the sodium gradient produced by the sodium-potassium pump as a source of energy to power the movement of glucose into the cell. In this system, both glucose and sodium bind to the transport protein, which allows sodium to pass into the cell down its concentration gradient, capturing the energy and using it to move glucose into the cell. In this kind of co-transport, both molecules are moving in the same direction across the membrane. Therefore, the transporter is a symporter. In a related process called countertransport, the inward movement of sodium is coupled with the outward movement of another substance such as calcium or hydrogen ions. As in co-transport, both sodium and the upper substance bind to the same transport protein, which in this case is an antiporter. As the substances bind on opposite sides of the membrane, they are moved in opposite directions. In countertransport, the cell uses energy released as sodium moves down its concentration gradient into the cell to eject a substance against its concentration gradient. In both co-transport and countertransport, the potential energy in the concentration gradient of one molecule is used to transport another molecule against its concentration gradient. They differ only in the direction the second molecule moves relative to the first. So this is showing you all coupled transport. We're ending the chapter now. The lipid nature of cell membranes raises a second problem. The substances cells require for growth are mostly large polar molecules that cannot cross the hydrophobic barrier of a lipid bilayer it creates. How do these substances get into cells? Well, two processes are involved in this bulk transport: endocytosis and exocytosis. In endocytosis, the plasma membrane envelops food particles, and fluids. Cells use three major types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Figure 5.16 can show you this. Like active transport, these processes also require energy expenditure. If the material the cell takes in is particulate, such as an organism or other fragment of organic matter, the process is called phagocytosis, or another way, cell eating. If the material the cell takes in is liquid, the process is called pinocytosis, or said another way, cell drinking. Pinocytosis is common among animal cells. Mammalian egg cells, for example, nurse from surrounding cells. The nearby cells secrete nutrients, and the maturing egg cell takes up by pinocytosis. Virtually all eukaryotic cells carry out these kinds of endocytotic processes, trapping particles and extracellular fluid in vesicles and ingesting them. Endocytosis rates vary from one cell type to another. They can be surprisingly high. Some types of white blood cells ingest up to 25% of their cell volume each hour. Molecules are often transported into eukaryotic cells through receptor-mediated endocytosis. These molecules first bind to specific receptors on the plasma membrane. They have a conformation that fits snugly into the receptor, and then different cell types contain a characteristic battery of receptor types, each for a different kind of molecule in the membranes. The portion of the receptor molecule that lies on the inside of the membrane is trapped in an indented pit coated on the cytoplasmic side with the protein clathrin. Each pit acts like a molecular mousetrap, closing over to form an internal vesicle. When the right molecule enters the pit, so the trigger that releases the trap is binding of the properly fitted target molecule to the embedded receptor. When the binding occurs, the cell reacts by initiating endocytosis. The process is highly specific and very fast. The vesicle is now inside the cell, carrying its cargo. One type of molecule that is taken up by receptor-mediated endocytosis is low-density lipoprotein. LDL molecules bring cholesterol into the cell where it can be incorporated into membranes. Cholesterol plays a key role in determining the stiffness of the body's membranes. In human genetic disease such as familial hypercholesterolemia, the LDL receptors lack tails, so they are never fastened to the clathrin-coated pits, and as a result, they do not trigger vesicle formation. The cholesterol stays in the bloodstream of the affected individual, accumulating as lipid plaque buildup inside arteries, leading to heart attacks. Exocytosis. The reverse of endocytosis is exocytosis. It is that discharge of material from vesicles at the cell surface. In plant cells, exocytosis is an important means of exporting materials needed to construct the cell wall through the plasma membrane. Among protists, contractile vacuole discharge is considered a form of exocytosis. In animal cells, exocytosis provides a mechanism for secreting many hormones, neurotransmitters, digestive enzymes, and other substances. So the mechanisms for transport across cell membranes are summarized in Table 5.2. Please use Table 5.2 as you prepare for your test. So this shows endocytosis by way of phagocytosis, or cell eating, at the top in 5.18a, and it shows pinocytosis, or cell drinking, in Figure 5.18b here at the bottom. And lastly is exocytosis, that discharge or release of materials outside of the cell used by way of plants or by animals to secrete things such as digestive enzymes, neurotransmitters, or even hormones. This has been your instructor,